Yeah, he got his PhD from McGill, the biology department, in 2015, and then spent a couple of years as a postdoc at the Center for Limnology at the University of Wisconsin-Madison. And then another couple working as a research scientist at DFO before just pre-pandemic in 2019, just as it was beginning, he became a prof at Concordia. There's a bunch of cool work in quantitative ecology, detecting regime shifts, metapopulation, meta-community theory. Of course, and he's also a noted GAM evangelist.
Go around the corner and see.
Let me welcome Eric Pedersen.
Go and turn the corner by the other door and you're going to see.
Thank you, Jake. It is fantastic to be back. Yeah, it is a real honor to be part of here. So, the quantitative biology program hadn't actually started when, I think it was just starting when I was leaving. And it is fantastic to see this broader group of people here and this many people interested in quantitative ecology and biology. So this talk is in part a bit of a work in progress. This is reaching its final stages, but this is an idea that I've been developing over the last couple of years to try and deal with what I think is hopefully I can convince you by the end of this is a kind of a fundamental problem in meta-community ecology, metapopulation ecology, that we still really haven't solved. We kind of just dodged. And that's how do we define what a patch is? So it's actually interesting, this one kind of unified, this project's one of my personal favorites right now, just because it's kind of unified my interest in theoretical ecology, statistical ecology, so driving things from measurement, as well as connecting some of the ideas about dynamic systems with how do we manage large-scale applied systems? How do we manage spatially structured populations? So it's a long, complex title. It's okay if you don't understand everything at this start, but hopefully by the end of it, all these words will make sense. So I suspect a lot of people in this room are familiar with the concept of a metapopulation, but I'm going to quickly go over the basic ideas of this. So in ecology, we think of we typically model populations either as continuous in space, so there's movement and interactions are happening across about the same time scales, and movement might be through diffusion or dispersal or other methods, or we might measure things using metapopulations. So metapopulations are a discretization of space. We're breaking landscapes into patches. So the core assumptions, the metapopulation assumptions, are that first of all, we break the landscape into two different types. So there's patches, and then there's matrix. So I didn't choose the name matrix. That's sort of the terminology we're stuck with. So you'll have to try and keep track of when I'm talking about matrix being the habitat versus matrix being the mathematical term. But matrix is just the spaces between where we expect organisms don't live. We assume that within patches, individuals are well mixed, so that all individuals living in the same patch have roughly equal chances of interacting with each other. So we don't have to try to model within patch movement when we're trying to model population dynamics. And we assume that interaction patches occur via dispersal. That is, typically there's no interaction except an individual leaving a patch and moving to a new one. And these dispersal events are relatively infrequent compared to the time scale of births and deaths within the patch. That is, it's a timescale separation approach. We're trying to break timescales into things that happen in patch, where we can assume everything's well mixed, and things that happen between patches, which we model as dispersal. So this has a lot of appeal, and I know this plot just shows some potential, some suggested subpopulations for northern cod. Apologies for not turning out that well on this screen, but these circles are different suggested subpopulations of northern cod, and these links represent potential dispersal events between these. So this highlights part of why we use the metapopulation approach, because it's really practical from applied and management standpoint. So typically when we're managing organisms, so I spent a couple of years at Fisheries Oceans Canada working on applied large-scale population modeling and spatial temporal population modeling. One of the things we keep running into is you can't manage a community as a continuous thing. You can't set quotas based on continuous areas of space. You can't allocate, when you're trying to assess how a stock is doing, it's really difficult to do that continuously. So typically when we manage populations, we try to do it on a patch-by-patch basis. We're trying to break things into subpopulations, which makes sense. So this problem is fundamental not only for theoretical ecology, not only for ecological modeling, but also for ecological management. So trying to break nature at its joints, as it were, is a fundamental problem that we keep trying to solve. But one of the things that I realized as I was working these kind of problems in DFO is something I had not really thought about before, is that we don't really have a solid definition of what a patch is. So we define a patch as everything is well mixed, we don't really have a dynamical definition of what well-mixed means. At least in terms of something we could quantify and measure for going from a continuous model. We know the universe is continuous. If we want to go from a continuous model to a discrete model, we need to come up with a definition that says, okay, this is enough local interaction for these to be well-mixed. This is not enough interaction for these to be well-mixed. So this fundamental definition of what is well-mixed is something we've honestly just skirted past. If you go back to the earliest metapopulation literature, we typically assume there are a bunch of patches connected by dispersal. And we really haven't made this connection to say, okay, like how do we actually define these patches in the field? This really came up when I was working on modeling shrimp fisheries. So I did a lot of work on modeling spatial temporal dynamics of shrimp. So these are very small organisms. These northern shrimp are, well, they look big on screen. They're only about, 3 centimeters, about 3 centimeters long. They live in a high abundance, and they're very populous in this area. And this should appear as a much better color scheme, but I think the screen is a bit washing this out. So this probably looks better to folks online. So these individuals interact with each other via competition for food, via competition for space, via mating, and they interact locally. So they're competing for the same food in small areas. So that's what this represents. But we also know that they move over incredibly long spatial scales. So in fact, when larvae disperse, they can actually disperse hundreds or even thousands of kilometers down this coast. And we actually know that they can move within these areas at relatively decent timescales. So one of the things we tried to figure out is, okay, how do we, one of my jobs was, and just to give you a sense of scale here, this coastline, this is the tip of northern tip of Labrador, down to the southern tip of St. John's. This coastline is roughly the same length of the coastline of the United States from Maine to the southern tip of Florida. And we had about 3 people trying to build a population model for this whole, all the stocks in this area, and trying to manage all the stocks in this area in basically a single integrated population modeling framework. And they said, you got a couple years to do this, go ahead. So I was running the issue of, okay, one of the problems we had to solve was how do we actually break this into patches? And honestly, what I ended up coming up with was I'm not actually showing the discretization here, but what I'm coming up with is this idea of basically a pretty crappy discretization into space based on depth and similarity. But I've always bugged me that we couldn't find a better way to discretize this into patches. So we know that interactions occur between individuals locally. We also know that movement takes place across multiple spatial scales. So movement ecology has been a massively growing field. It's grown exponentially even in the time as I started as a PhD. And one of the things we've learned from it is that individuals, we've been tagging and tracking individuals. So this is a lobster that's been marked and recaptured over multiple times. We learned that this movement happens at multiple different scales. So typically individuals often spend a lot of the time moving over short distances. We also have individuals that can move over much longer steps. There's often intraspecific variation in movement. Some individuals move quite far, some individuals move short. And there's strong habitat preferences. So individuals don't just move following some sort of diffusion process. They typically have strong preferences for specific types of habitat, specific areas that they tend to move through. So we want to ideally take that into account, the fact that movement patterns into account when we're building patches. So I think part of why we initially didn't really get into this issue about what a patch is as ecologists, because the first metapopulation models were typically applied to things that made sense as patches. Island biogeography, where islands are a very natural patch. You could assume things in an island interact with themselves, things between islands interact with each other. Or as another example, Ilka Hansky's initial work was on patches of meadow and forest. So these are openings in the forest where butterflies are moving in the patch, and they typically are not going to leave that patch frequently except to move between them. There's a pretty clear view of what a patch is. So we never really had to encounter or deal with this issue in the early metapopulation models. And a lot of times, actually, we've kind of just gone by saying, okay, in continuous landscapes, we're just going to treat patches as any contiguous chunk of habitat. However, what we run into with continuous space, though, is that, or spatially continuous systems, is that we rarely actually get discrete contiguous chunks of habitat easily broken apart. So this shows a plot of a habitat, so what's called a species distribution model for northern shrimp, showing areas where we expect to see high and low biomass density. And you can see this is patchy. There are areas where you have very high density, you got areas of relatively low density in between, but there's no actual discrete breaks in here. So you squint at this, you might be able to draw circles around some of these and say, okay, that might be a patch, but we want ideally a more rigorous way of defining this. And this just shows sort of a map of habitat quality, so the implied habitat quality from this map. So it gives you a better picture of areas of high and low expected occupancy. So this really raises the problems. The problem I wanted to focus on then is, can we get fundamentally back to this idea of how do we actually define what we should consider to be a patch in a continuous landscape? And to get into the headspace of this problem, I want to actually do an exercise. So hopefully everyone's paying attention, because this will be interactive. I've been teaching undergrad classes enough that I've gotten very into interactive questioning. So imagine we have a landscape like this, where we've got black represents high quality habitat. This is core habitat, reproduction, food is all available there. Gray areas represent moderate quality habitat. Individuals will move through those areas and will still spend time residing there, but don't necessarily breed frequently in there. And then white represents areas where individuals will move through, but when they're moving through, they move through rapidly, and they don't linger there. So given what I just told you, how many people would consider this to be two patches? So just raise your hands if you think this is two patches. Okay, pretty much everyone. How many people consider this to be one patch? Okay, not. It depends. I like that. Well, yeah, you're spoiling my story here. Okay, so let's try a bit more tricky one. So everyone can agree that one's a patch. What about this one? How many people would agree that? Who many people would say this is two patches? Okay, how many people say this is one patch? You got to put up your hand for one of these. So, okay, so mostly people are saying one patch, a few two patches. What about this case? So we've got two core areas with a very fine dividing line between them. So very little space. How many people say this is two patches? Okay, maybe about half. How many people say this is one patch? Maybe about half. Okay, so as Fred pointed out, this is a trick question, because whether this is two patches or one patch is really going to depend on the organism itself that's moving through this landscape. So if you're a snake that moves through habitat, doesn't necessarily have a strong home range or habitat preference, but hates moving through open ground that might contain birds, might contain predators, and wants to stay in cover, this is probably one patch. They can move through this pretty easily. They can move rapidly. But if you're a bird that tends to stay near core habitats and might not travel far out of like these ideal ranges, this might functionally be two patches. So birds in this one might be different from this. So depending on we're talking about birds or snakes, these might be two. And the opposite would be true for here, where birds could very easily move between these two patches. So their core areas, they would be able to move back and forth and use those same resources. But a snake would be terrified to go. A snake might never make this small distance because it's open ground that's opening them up for predation. So we want to define what a patch is. We need to take into account how the organism is moving through the landscape. We can't just look at contiguous spaces. We have to look at movement. So I argue that we should be defining patches based on scales of population synchrony. So something should be considered a patch if there's sufficient intraspecific interaction within the patch that it fully synchronizes the dynamics. Basically, that maybe in simpler terms, we expect that individuals in one side of a patch should have very similar dynamics. Like the population dynamics should be pretty similar depending on which side of the patch you are. You can't tell where you're in the patch based on looking at the abundance fluctuations around you. And just to give you an example for this, and I obviously couldn't be one of Fred's students if I didn't at least bring synchrony once into this talk. But this is definitely part of what we were thinking about. This is trying to define this, because if we look at these different areas, so these are three major channels where shrimp were initially found. So these are kind of core areas for shrimp where they actually occurred in the 80s before the shrimp expanded out to this whole region. And what we find is these three channels, even though they're close in space, have very asynchronous dynamics. So this is the hawk channel here. This corresponds to this one. You can see it massively increased and collapsed. And then this is Hopedale and Cartwright. So these are both slightly more north. We see they both showed they showed an increase, but not nearly the same extent. We also see that they have relatively synchronized dynamics. So not exactly synchronized, but closer. So if we were making a first pass of what might constitute patches, we might consider Hopedale and Cartwright to be in the same patch, or at least closer to each other, than we might consider hawk to be. Now, however, this kind of pushes the problem one step further, because it's saying, okay, we can define it based on synchrony, but synchrony, if we want to try and measure it in the field across a whole area of space, requires a massive amount of time series, a massive amount of effort, and for practically any species, we can't do this. We can't actually measure synchrony directly as much as we'd like to for most species in most situations. However, we can base this idea, based on the idea that synchronization occurs more rapidly when there's strong interactions. So there's a lot of so if you have individuals that start out at either end of the patch are likely to bump into each other in their lifespan. So if mobile individuals are likely to encounter each other at different parts of the same location, well, they're likely to interact and likely to synchronize dynamics. So we can kind of go one step forward from synchrony to say, okay, for mobile species, can we define this based on probabilities of interaction? So this basic project is focusing then on, okay, if we have a movement model, so if you're given by God a model of a movement for an individual, can we use that model to infer what rates of interaction might be? And we use this to define what patches should be. And I think this focus on interaction, focus on movement, gives us at least a couple of these qualitative ideas about how we can start to define what a patch should be rigorously. So first of all, an area. So an area should be considered a patch. So I'm drawing, so this is, just an example habitat here. So black represents high quality, gray represents middle quality, and white represents low quality. And we have a few interesting cases. Actually, I'm going to focus on this quickly just to highlight. So this might or might not be two patches by that same criteria, depending on the organisms move through it, because it's got two high quality areas separated by a mid quality. And this one similarly might be one or two patches because we've got a couple high quality areas connected by a thin area between. So you might ask, okay, focusing on this area, is this two patches or one patch? And should depend on the animals or the organism's movement. So maybe the basic rule should be that if something is a patch, if we draw an area around someplace in the map, that's an area where individuals should be likely to be found. So if the species in the population, if there's population present in the landscape, a patch should have a high chance of finding an individual, and if individuals that's inside that patch should have a low probability of leaving that patch. This is the most maybe fundamental idea of what defines a patch. So this one shows a random walk of a red individual moving, and you'll see this individual starts out at this point and moves around this area and does actually one long distance displacement to here, but still doesn't switch back. So it's still spending its area. So we might be able to think of this, okay, these are probably different patches because individuals are not frequently leaving either area. But that on its own is not sufficient because we have to then decide where we draw the lines between patches. So the next idea then should be that two individuals in the same patch. So if you define the area again, an arbitrary area as a patch, if you pick any 2 points within that patch, individuals that start out at those two different points should be roughly as likely to interact with each other as they are with another two individuals that start out from the exact same site in that patch. So that this is, kind of trying to give some teeth to the idea of this of well mixed. That if you look at a sufficiently long time if you know where two individuals start, you look at a sufficiently long time, if they're within the same patch, you shouldn't be able to predict you shouldn't be able to tell which one should be where in the patch after that time horizon. They should be basically anywhere within it. So the nice thing with this, though, is that this actually gives us the grounds for building a distance metric, a way of measuring distance based on interaction. So we can think of the frequency of interactions between individuals as a measure of distance. So if you look at this, so this is showing the same landscape. So this is the continuous landscape on the left. This is a discretized version of the landscape on the right. Make sure I'm conferring this right. Just showing it blocky. And this is showing individuals that's starting on three different locations. So individual 1, individual 2, and individual 3. So these are three different starting locations. We want to see, okay, how frequently the individuals encounter each other given they started out in these three different locations. So the right-hand side shows simulations of 20 different movement paths from a simple movement model. Basically, the movement model is individuals tend to move from mid to high. They tend to move from low to mid or to high if they can. There's a finite distance of dispersal. And when they're in low quality habitat, they tend to move quickly in straight lines. If they're in higher quality habitat, they tend to move shorter distance and more sort of cyclical motion. So given this movement model, we have a case of, okay, individuals that start on 3 here, most of these individuals are all moving around in this area, whereas individuals 1 and 2, they actually do a decent job of kind of exploring this space. You notice that this area doesn't actually get visited that frequently, so this is a bit lower priority. But notice that even individuals that start out here, they still end up encountering each other in this place. So even though they start on different locations, they still end up encountering each other relatively frequently. So if we did this simulation many, many times, so we did like not only just 20, but if we did a whole number of paths and actually counted how many times they encountered each other in different simulations, and then looked at the average number of encounter rates over time, we'd have something that looks like this. So if we define I, so capital I, little j, this is our interaction rate for two individuals, or sorry, two different sites. This gives us the average number of interactions per unit time, so averaged over some span of time. The average number of interactions between individuals that start out in patch I and patch j. So the plot on the left shows these average number of interactions. So this is, if we look at longer, if we look at only a very short time horizon, obviously individuals are interacting with each other quite frequently. So this actually grows because individuals are bumping into each other quite frequently when they start out, but eventually they'll move and mix in the area, so we start to see a slump. So eventually, over a long enough time horizon, all of these total interactions will actually go to a constant across this landscape. So an infinitely long time horizon, this will go, all these lines will eventually drop to a constant, but how quickly they drop to a constant gives us a sense of like how often they send bumping into each other over short time periods, like demographically relevant time periods. And the plot on the right here shows the rate of interactions between individuals from different sites. So if you go back here, remember site 1 and 2 are quite close, they're in this area. So these might be different patches, they might be the same patch. You see site 1 and 2 bump into each other quite frequently, whereas individuals from site 1 and 3 or site 2 and 3 bump into each other very rarely. So they're not likely to encounter each other frequently in any given span of time. So this still gives us, this still gives us the problem of how do we actually turn this into a distance, so something we can actually, that's symmetrical between patches. We can actually define a distance based on this by taking the sum of self-interactions within, so this is interactions within the site, plus for site I, interactions between individuals for site j, minus 2 times the number of interactions between individuals in site I and j. So this actually acts formally as a distance. So it's bounded, below by 0, and Noe, and so this is based on some work by Noe and Clementi, a couple of mathematicians working on Markov random chains in molecular systems. One of the things to show that this actually has the properties of a distance metric. So we can actually use this as a distance for measuring encounter rates between different states in a complex system. And we can see we actually get a distance metric that over time, we see that over short time horizons, the distance between I and 1 and 2 and 1 and 3 grows. The distance between site 2 and 3 doesn't grow that quickly. And again, eventually this distance will eventually go to 0. So over an infinitely long time horizon, all these distances will shrink to 0, but it's the finite time horizons, of course. You want to see number of interactions in a reasonable amount of time. And we can see that, okay, site 1 and 2, sorry, I think this is mislabeled. So this should be site 1 and 2. So this should have a relatively short distance, and then these two are going to be further from each other. Apologies, I think I messed up the labeling on this slide. Okay. So this does give us then a metric for considering areas. And we could actually take, you could take any movement model you wanted, simulate it across a big enough landscape, and count the number of interactions. So if you have a way of simulating interactions, we could do this in principle for any movement model we want. However, this is computationally insanely intensive. We'd have to simulate many individuals moving from every single site and count the number of times they interact with each other with every other individual from every other site. So that's computationally incredibly expensive and probably not practical. But can we do this efficiently in a way that we can actually compute this in finite time, or at least reasonable time? And so it turns out we can, as long as we're willing to make some simplifying assumptions about the movement model. We restrict ourselves to specific sets of movement models with specific properties. And we actually, the restriction we have to put on here is what's called, we have to assume a Markov movement model. So I'm focusing here on what are called continuous time Markov chains, because it's actually easier to think about these in this. So these are individuals that, so this is a movement model where individuals start out at a given site, and then they wait some amount of time before moving. So there's a random distribution, exponential distribution for how long they wait in the patch. Then they move. That's patch dependent. And when they move, they pick a site in a neighboring area with probabilities based on the relative characteristics of the site. So this actually allows us to model continuous time movement very easily. It also allows us to incorporate things like habitat quality, distance, relative habitat quality of two sites. So it's not just dependent on where you're moving into, it can also depend on where you're moving from. And also allows us to do things like take into account movement distance. So if some individuals might move quickly or slowly in some landscapes. So given this, we assume that there's some probability, Pij, at time t plus some distance tau. Distance in time tau. So this represents, okay, the probability of finding an individual that started in grid cell I at time t, ending up in grid cell j after time t. So this is basically a probability. And if we make the assumptions of stationarity in time and stationary in time, so that is, if you started at a given patch, that's all the information you need to predict the individual's future path. So there's no changes in movement preferences over time, and there's no memory. So individuals don't remember the path they took, but they can evaluate the habitat around them when they're choosing the next step to take. So given these two assumptions of stationarity of time and only dependence on habitat, rather than dependence on history, we can actually simplify this down to a single probability that only depends on the separation in time between two time points. That is, if we know the current state of the population, we can use that to interpolate forward. You don't actually need to know the starting point of when the simulation did, as long as you know the current state of where individuals are on the landscape. This is very nice from a mathematical standpoint. I'm not going to talk too much about the details here. This is really just to show that we've done the homework. But one of the nice things, and to hopefully get people interested in continuous time Markov chains, because they are actually a very powerful tool for modeling movement. And one of the nice things with them is because of these assumptions, so if we have a matrix function of tau, so this is again our function. So given the starting probability, given starting time 0, after tau time, what's the probability of individuals moving from different sites to different sites? So given these assumptions, we can actually form this as a linear dynamical system. So we can say how these probabilities should change over time. It's a linear system that depends only on capital M, which is a movement matrix that describes sort of the habitat relative preferences between different locations in the landscape. So if you've got n sites, this is an n by n matrix, and it depends on the current state, so the current probability distribution of where individuals are in the landscape. So what we can do then is actually solve this for any amount of time. So we can actually pick an arbitrary time scale and solve this, and find that we can actually solve this as a solution of the matrix exponential of this times our initial distribution of individuals across the landscape. So we know where the individuals started out. We can use that to predict the probability of finding them on the landscape at different points in time. And then the movement matrix here, it depends on the habitat characteristics and the distances between sites, but again, it doesn't change over time. We're not assuming time variation. And this system is actually conservative. So if you start out with, say, 50 individuals on the landscape, run this model forward in time, you'll end up with 50 individuals on that same landscape at any point in time. There's no births, there's no deaths. We're just assuming only movement in this model. So given this, then, we can actually simplify our problem from trying to simulate, movement from n different sites across, say, multiple time starting doing multiple replicates, which would be an incredibly difficult problem, to one that's much more easily solvable based on using only information stored in this movement matrix. So this is the basis of what we've been calling the eigenpatch algorithm. So this is work I've been doing with an undergraduate, actually just now graduated, but he's been in the lab for about 3 years now, Spencer Sherman. So we've been working together both on developing the theory for this and developing the Spencer's done a ton of work on the computational side, so actually applying this to more complex landscapes and working out some of the bugs of the code. The basic idea of this algorithm is first, if we start with a continuous time Markov chain model, so these are actually relatively straight, this is a relatively popular model, becoming more popular now for estimating movement. And there are actually a wide range of applications where we can actually estimate these movement models from paths of data. So assuming you've been given continuous time Markov chain movement model, we use it to generate our movement matrix M. So that's the probability of moving from each of these sites to each other site. The derivatives of those probabilities of moving from each site to each other site. And then this is our first step for simplification. So, if you remember, if we wanted to actually calculate those interaction probabilities, we could do it by doing that matrix exponential at any given time step, turning in probabilities, multiplying it out. But solving a matrix exponential for an n by n matrix, which is what we have to solve here, is not computationally cheap. It's like, it'd be n to the power 6 operations or something similar. So it'd be an incredibly difficult and expensive operation to do. And it's not something we could feasibly do for any reasonable landscape. So what we do, first of all, is actually try to approximate this matrix by finding the first D eigenvalues. So these are basically like we're finding a reduced version of this matrix is only using the first D eigenvalues and eigenvectors from it. So lambda 1 to D being the eigenvalues. And then we take the right and left eigenvectors. So phi here being eigenvectors, psi being eigenvalues of M. left eigenvectors. Whoever invented the terminology for eigenvectors, we need to have a discussion, probably in German, because so this gives us basically a way of approximating that movement matrix. Basically, we can use it to approximate the movement at any given time. I'm not going to show the math here, but we can actually use these D eigenvectors and eigenvalues to come up with an approximate value of the probability of finding any individuals. It's computationally much, much, much cheaper. We typically found we only need about 30 eigenvectors to do this effectively for a landscape of 1,000 by 1,000. So that's shrinking our computational size from 1,000 by 1,000 to a 30 by 1,000 calculation, which is incredibly time efficient. So once we have these eigenvectors, we can actually use this to, first of all, we can then calculate the landscape. We can use this to break it up to be our first rule. Remember, our first rule is individuals who tend to stay in patches. We should calculate something as patches of an area of high occupancy. So this leading eigenvector, the leading eigenvector associated with the eigenvalue, actually splits the landscape into areas of high versus low occupancy. So the value is high, you're very likely to find an individual over long term, like over an infinitely long time horizon. These are the places you'd find an individual wandering in this landscape. So this gives us our first criteria to split things into patch versus non-patch. So we can always classify the landscape into, we can always basically throw out a lot of the information on the matrix here, because all we need to know is about movement in these patches. Then the next step is actually using the remaining right and left eigenvectors to calculate an approximate kinetic distance. And this figure turned out very bad, but this is actually showing a distance of this point to all the other points. So this kinetic distance being the probability of this scaled probability of interaction. And this delta ij represents, basically it's like xi is a probability where individuals start out in all the individuals start in patch I. Xj is initially all the individuals start in patch J. This delta xij is, okay, this is the difference between patch I and J. So this actually lets us codify this interaction probability. And then we just do this kind of complex matrix, which I'm not really going to dig into because this is already overly complicated and I'm getting tired of Greek notation. But you hopefully take my word for it. This is a relatively simple adaptation of an eigen decomposition of this matrix. So if you know what that means, that's great. If not, basically we've just done something that takes an incredibly complex computational problem and solves it in much, much simpler time with very little information given up. And that gives us now a matrix that we can use to cluster our locations in space. So the final step is then using this distance matrix and applying whatever your favorite distance algorithm is. So we've been doing a lot of work on this. Spencer has found that there's an algorithm called dbScan that we've been using that works on multiple scales and actually works effectively for clustering. We're still trying to figure out the best, most appropriate clustering algorithm, because how you cluster this, the algorithm used to cluster this is almost as important as distance matrix in determining what clusters you get. But if we apply this dbScan algorithm, we actually get a sensible cluster. So based on our encounter rates, we actually find that this yellow area is separating into a separate patch from the orange area, even though they're physically connected. Whereas the green area, which remember had two high quality areas, is connected into a single patch. So this takes the movement model and actually gives us a reasonable way of finding the patches without us having to say, this is what our threshold for patches are, or this is what we consider to be contiguous or not. And actually getting back to my original motivations for this, I wanted to try to apply this to Northern Shrimp just to see if we can. And basically as a practical application to show this works in a real system, not with just a simple toy model. So in this case, I use the eigenpatch algorithm. So first of all, we estimated a movement matrix M based on some simple movement rules. So we don't actually know how shrimp move. We have some idea, but we don't have detailed knowledge of how they move. It's really hard to put a radio collar on a shrimp that lives about 500 meters at the bottom of the ocean. So because of that, we do know some general characteristics about how they move. We know they don't typically move as adults more than a few hundred kilometers in a year. We know that they typically prefer to move to specific depths. So there's specific depths they prefer. So they'll typically move from a shallow area to a deeper area, or to these preferred areas. And we know that they, we suspect many marine organisms will typically show this pattern, or they typically show like relatively straight line movements in areas of low quality habitat. So they're in low quality habitat, they'll typically try to make a beeline in any direction to find some high quality. We don't know if this is true or not. We'd love to do some camera tests or something to see if it works. But these are sort of the assumptions. What we can do is we can actually use this to estimate a movement model that tries to best fit this distribution of biomass density. So we try to tweak these parameters. We basically use an optimization algorithm to tweak these parameters to find a movement model that should recreate this map. And then given that, we can actually use this, we can apply the movement model. We end up with a pretty sensible set of patches. So we end up with, so these are showing the patches here. The colors are very pastel-y here. I kind of like it. It's very more appropriate for Easter than for Halloween. But these showing different areas of patches. So you'll notice that these areas of contiguous habitat here are actually getting split into separate habitats. And so these red borders actually show our management areas. So these red borders were drawn pretty arbitrarily to how to break this into areas to manage back in the 90s. But one of the things we always suspected was this is SFA7. This is a southern shrimp fishing area. This is SFA6, one of the most major ones. One of the things we saw looking at synchrony of dynamics was that these populations, there was a big collapse in these populations, and that collapse started in SFA6 and then propagated to SFA7. So we saw SFA7 collapse, and it collapsed much harder than SFA6. And part of what we think was happening is that this SFA6 is, there's basically a big southern current that's sweeping larvae out. And like, this is not a lot of ideal habitat. So we think that this is actually basically a sink habitat, that these individuals are only able to, you only get a shrimp population here because they're being subsidized by movement from parts north. This is actually well supported here, where we're seeing that these two patches span this border and with a large amount of area. So we expect that if we see a decline here, we would also expect to see a decline here. And notice that this is a very abundant area of shrimp. So we expect if we saw an area of decline in our most abundant area in the north, we'd expect to also see a decline in the connected area to the south. So hopefully this is, and this is still the preliminary steps of this. We don't necessarily expect to directly apply these patches to this system, but this does at least give us first steps to hopefully try to start dividing this into more reasonable areas to actually model and manage this fishery. So just briefly summary, our basic argument is we should be defining patches based on encounter rates, not just continuity of habitat. And the second is that we have this eigenpatch algorithm that we can use to model movement. So as long as you're willing to accept some constraints on how you're modeling movement, we can simplify this down to a much simpler problem and actually solve this in a reasonable amount of time. So actually, and just a bit of a, so this is like, I think this is something like 20,000 squares. So this is, if you think about the actual dispersal matrix we're trying to estimate here, something like 20,000 by 20,000 dispersal matrix. That's a pretty big matrix to try and solve by standard matrix exponentials. We can actually calculate the distances here in about a minute and a half. And there's still a lot of numerical optimization here. I think I can probably get that down to maybe 20 seconds. So this is hopefully trying to demonstrate that this is not just something that can be applied to only toy models. This is already a practical algorithm that we can apply to complex, large-scale systems that we want to use this on. So where are we going next with this? So first of all, I realize I think I got these out of order. So first of all, what we want to do is actually do this dynamically. We want to compare clustering calculations. So looking at these clusters and see if they actually predict synchrony in a toy model. So actually, if anyone on the dynamic side who's looking for an interesting project or interesting challenge to take on, I'm very happy to talk about this, because this is something we're looking for collaborators on. So trying to actually compare population dynamic simulations with our clustering algorithm results. We want to try to find better default settings for the clustering algorithm. So we're still trying to figure out the best clustering algorithms that give us consistent and repeatable clusters. And then finally, so that's what the pictures on the right here are. So this is Spencer, and then this is Allegra Spencieri. So she's a master's student in the lab who's been here she's in her second year now. And she's actually applying this, so she's collecting data to apply this method on butterfly movement. So I am a quantitative fisheries lab, but we decided that butterflies were both, easier and cheaper to work with than shrimp for our initial test. So we are now studying what Allegra calls the fish of the land. So we're looking at the movement of European common butterflies and trying to use this to parameterize these movement models, and from there try to understand the spread of the species. So this is European common blue here. This is a relatively newly arrived non-native and invasive species. It's spreading quite rapidly through Montreal and Toronto, across sort of the Quebec-Windsor corridor. We want to understand, okay, what might be habitat, like what might qualify as actual habitat patches for the species. So Allegros is trying to apply this method to see if we can actually use this in a practical setting to infer habitat patches for real species. So I'll quickly say thanks to all the folks in the Pedersen Quantitative Fisheries Lab, as well as a bunch of people who have given me useful feedback over this. So Justin Marlowe, just for frequent discussions and conversation of this. Melissa Guzman for initial conversation and some of the early ideas of this, as well as Patrick Thompson. And Fritjof Lucher, who I think I saw online, so hi Fritjof, who gave me a lot of useful feedback on solving kind of some tricky matrix problems over at ESA last year. Yeah, very thankful for everyone. And if you're interested in the lab's work, you can follow us on Twitter. If you have now given up on the Hell site, you're also welcome to follow me on Blue Sky if you're on there now. It's the same handle, and I'll still be tweeting lab work there. And we also are at the Pedersen Fisheries Lab. And I'm happy to take any questions. Thanks for your time. Yeah. Well, thank you.
We have a PhD student in our lab